Okay, good evening everybody, warm welcome. I'm Tom Wilkinson, I'm one of Professors of Respiratory Medicine, and I've been asked by Rox Crewe, who's our head of school in CS, to introduce this evening's lecture by Professor Karl Staples. So I have known Karl now for 16 years. I was checking my records, having moved here in 2007, a year after Karl started in Southampton. So I think I'm well qualified to understand the man in question. He's a wonderful partner to work with in research. He is a generous person, and we're going to hear a lot about his journey this evening. But I just wanted to really highlight some of his traits and really introduce the theme of respiratory research to you all, who many of you know it well and others not so. So Karl started his academic career in undergraduate studies at Surrey, and then moved to really one of the leading centres at the time, along with Southampton, the Royal Brompton Hospital, and worked with Sir Peter Barnes, who was a leading researcher in COPD and infection, and then travelled a bit to Leicester and New York, and eventually came here to Southampton, where he's made his career. And I met him a year after starting here. We shared an office, a very small office, under Ratko Djukanović's mentorship, and had a really wonderful time sharing ideas and creating some really interesting discoveries, of which Karl was the key component of those. He's a very generous person with his time, and I think his main legacy here is actually the culture change he's enabled for junior researchers, for the early career researchers, many of whom have come today, having left years ago, which I think is testament to that. He's worked in the doctoral research community, in supporting postdoctoral researchers, recently in establishing a whole way of working within BRC to ensure there's a diverse and equitable approach to fellowships and supporting new research careers. His work is internationally reputed. He's recently elected indeed as chair of the British Association of Lung Research, which sits at the pinnacle of respiratory research in the UK, and we're really proud to have him as a member of our research community in Southampton. So it's a personal, real honour, Karl, to introduce you today. Look forward to hearing about your journey, and we thank you very much for your work and attention. Thank you.
Wow, wasn't expecting that. So good evening everybody, and welcome to my uncertain journey through a career in respiratory medicine. It's uncertain because at many points in this journey you will see that there were different paths I could have taken. Some of them could have been more attractive, who knows, because I didn't take them. But there were forks in the road that were evident, and I'm going to share some of those with you this evening. So, this is who I am. This is what my CV... Sorry, no, I'm in disguise. This is my current academic CV, which looks, on paper, very, as though I was on an upward trajectory all of the time. This is a lie. There are lots of points throughout this journey where things didn't go the way one would have planned or expected. And I'm going to highlight some of those, along with all of those mentors who've helped me along the way, and all of those opportunities that I have taken to basically have fun with my career. Because if you can't have fun with your job, then I think you need to look for a different job. So, in the beginning, for those of you who know me, and I know that some of you online will recognise me stuffing my face. This is a theme that's happened throughout my life, and indeed this apparently was the first picture of me eating an ice cream. So I was I've always enjoyed my food and continue to do so. The middle picture, when I just started primary school, I haven't lost my teeth as a result of eating too much ice cream. That's me when I've lost my teeth, and I'm also suffering from chicken pox at the time. But when I reached senior school, that's when you start to get pigeonholed, and when you start to learn about yourself and what you enjoy doing. So that's me on the picture on the right there, where I was very involved at school with filming events, so video cameras, photography, that sort of stuff. And there I am doing a live edit of the school sports day. Notice how our technology has changed throughout the years. I mean, how I ever carried one of those TVs, I don't know, because it looks bigger than me. But at school, I enjoyed science. And I got the reputation, as one does at school, of being a nerd, a geek, a boffin. I didn't mind that, because I really enjoyed what I was doing. Reason I was a nerd, a geek and a boffin was because of all of these early influences. Those of you who have looked at that first slide where I put some dates and have done some quick maths may be able to work out how old I am. However, I'm sure these pictures confirm your suspicions. So we have Johnny Ball and Think of a Number, Johnny Morris, David Attenborough, of course, Sir David Attenborough, David Bellamy. All of these people were on TV at the time doing educational programming, showing us about the natural world, how the natural world works. And I really enjoyed these programs. I really enjoyed finding out. And what I liked was that I would find out facts. At the time, I was a fact fountain. I really enjoyed just knowing stuff. However, it wasn't until I was slightly older when my mother... Hello, Mum, by the way. Sorry, I should have said hello, Mum, before. Apologies. And you may recognise this gentleman here, Quincy, medical examiner. He was a forensic pathologist, very much before CSI and all these fancy machines and all the things where they really annoy me when they put them on TV, because none of those machines work like that. But anyway, Quincy ME, and this was really my first introduction to show that science wasn't just about knowing facts. It was about science being used for a reason. Obviously the reason was here to solve crimes. There's also obviously a picture of the Star Trek crew here, one to even further confirm that I'm a nerd, but also because they used science, obviously far future science, again, to solve problems. And this was why I really enjoyed doing science. But it was really Quincy that captivated me, and what I wanted to be when I grew up was a forensic scientist. And as I say, that's one of the reasons some of these CSI programs really annoy me now, because I know a little bit about it. So, how do you get to be a forensic scientist? At the time, there were only 2 places in the country that did it. There was the Metropolitan Police Forensic Science Laboratory, based in London, and the Forensic Science Service, which had different hubs throughout the country, but their main lab was in the Birmingham area. But how did you get into this? Well, somebody said, why don't you go to university? What is university, I said. It wasn't on my radar. I didn't know about university. I am the first person in my family to go to university because it wasn't really on our radars. We didn't know about it. So, when somebody said, yes, go to university, do a degree in biochemistry. Okay. So, you did the usual tours, went to lots of universities, visited them all, and I went and chose Surrey. So at the time, the University of Surrey was offering a biochemistry degree in toxicology, so drugs and poisons. That's how I always told everybody what I was doing. But the reason I chose them is because they had good links with the Metropolitan Police Forensic Science Laboratory and with the Forensic Science Service. Because at Surrey, a key part of their undergraduate programme was a third year in industry, so a professional training year, where I went out to get some real-life laboratory experience. So again, I'm sorry, another picture of me stuffing my face. But when I got to the point, I was lucky enough to get a place at the Forensic Science Service in Birmingham, looking at new short tandem repeat loci for using in DNA fingerprinting, which at the time is cutting edge. We were using PCR. So for those of you online who don't know what PCR is, polymerase chain reaction is where we can take DNA, amplify it, so we've got enough to actually do some analysis with it. So this was how you could take small amounts of DNA from a crime scene, amplify it up, and so you could tell whose DNA it was. This was looking at very small parts of the DNA that were regions that were hypervariable. They differed between people. Now we can sequence the whole genome, and we can do it with very small amounts of DNA. But at the time, this was our cutting-edge technology. And I was even proud that I could print stuff out in colour on a laser printer. But yes, so this picture here was of a gel that's been actually analysed using a laser. But what I learned whilst doing this research at the Forensic Science Service was that at the time, again, only 2 laboratories in the country. They are civil service jobs, and they are very, very hard to get into. Indeed, at the time, somebody told me, oh no, you'll not get a job here, it's dead man's shoes. Because it was all about somebody went in and they spent their life in the civil service and they left. Nowadays, on the other hand, these two places no longer exist, and there is a market. So there's now lots of jobs and also lots of degrees, you'll notice, in forensic science. But what I did learn through that year was that I enjoyed research. The ability to come in, learn something new by applying the science that you did to a problem, generating new data and finding things out. You know, that sort of detective work that was evident from my early interest in Quincy Medical Examiner. So, at the end of my time, I did graduate. Hooray! And so I decided then, well, how do I stay in research? What am I going to do? And everybody said, oh no, to stay in research, you've got to do a PhD. What's a PhD? So obviously I then went and did some research, because I'd now been trained in it, and learned that PhD was a doctoral training program where you learned how to do research and how to apply research. So, this is the first fork I'm going to introduce you to in this lecture. So, it's called Piled Higher and Deeper, or the PhD. I've stolen that, obviously, from the comic, so apologies to anybody online who's trying to sue me at the moment. But this fork, because I applied for and was awarded a PhD in toxicology. And this PhD in toxicology was looking at the expression of a gene called erythropoietin, which was the main molecule responsible for the production of red blood cells, and how it's controlled by oxygen. This, at this time, was the hot topic. People have now won Nobel Prizes for knowing how oxygen is sensed molecularly within the body. Why is it a fork? Well, I was there 3 months, and they said, we're going to move labs. Fine, that sounds good. We're going to a new building. And I subsequently found out that the building we were in, Professor Wilkinson joined us went straight into the building as we moved out. So he's been following me around all of his career. But yes, so we're going to move labs. Brilliant. So we get everything packed up, we move over to the new labs, and then commissioning the new labs took another 5 months. Great. We're just about to now start with doing some experiments. We're being put out to tender. Sorry, I should have said. I was doing it in the Department of Health, Department of Toxicology at Bart's Hospital at the time. But the Department of Health wanted to put it out to tender. But don't worry, we'll be fine. Lost the tender, it went to Imperial College. So, 15 months into my PhD, I had to make a decision. Why am I doing a PhD? Do I still want to do a PhD? What is it I want to gain from a PhD? There was a lot of soul searching and possibly some alcohol involved. But at the end of the day, I spoke to myself and decided that yes, I did want to do research. I want to use science to achieve something. So that led me to my second PhD, obviously not awarded, but looking at the role of the immune system and how the lungs work, and specifically in lung disease. So I'm now going to take, if I may, a small diversion into some biology. These are the lungs. I hope you, at least some of you recognise them, at least some of my students at least. We're dividing them functionally. We've basically got the conducting zone, which is basically, for those of you online, the plumbing. This is how we get air to the place it needs to be, which is the respiratory zone. And this respiratory zone is where the alveolar spaces is, and this is where we've got a thin lining of cells which are so thin that gas can easily cross between the air in the lungs and the blood stream. So that's what it's all about. The lungs are there to get oxygen into the blood and get rid of carbon dioxide from the blood and out. Now, when you look at some lungs, they look fairly homogeneous. They look as though they're one thing. But they are not. And this diagram on the right-hand side here just shows you the cellular complexity just in the airway parts. And this is just in the parts of the lung that are exposed to the air. We've got epithelial cells, which are really the barrier cells, but they also perform functions. They also move things. They have cilia, these little fine hair-like projections on top of the cells, and they are able to move the mucus on these so-called mucociliary escalator up from the lungs into your throat, so they can either be swallowed or expectorated. But there are differences even in the epithelial cells, and they change as you move down the airway. Obviously, the ones right at the end in the alveolar sacs are the most specialised. They are thin, and they allow that gas to move from the air into the blood. But you need a lot of different cell types to get there. More importantly, these are just the ones exposed to the air. There are lots of cells underneath in this so-called subepithelial layer, and the submucosal layer. There are lots of different cells here too, such as smooth muscle cells. And these smooth muscle cells are the contractile cells that are really responsible for that bronchoconstriction, that twitchiness that we see in diseases like asthma. So, speaking of the respiratory diseases we'll be looking at today, we have asthma, which is the most common chronic disease in childhood. Again, these diseases are all relevant and all impact not only on the NHS, but also on the people that suffer from these diseases. So the direct asthma cost in just people going to hospital and having drugs is £1 billion per year. But most importantly, despite therapy, this disease does still kill people. And so asthma, we sort of concentrate on the earlier years. I mean, I know, and Ramesh is in the audience, and he's now looking at asthma patients who are moving towards the older end of the spectrum. But we've also now got chronic obstructive pulmonary disease, COPD. It's a disease that's mainly associated with smoking and older age. Unfortunately, with both of these diseases, they are syndromes. They are syndromic in nature, and that really just means we are only diagnosing them based on symptomology and the patient history. We've got very few specific molecular tests to tell us what sort of underlying disease somebody is suffering with. And even now, we're coming up against diseases like asthma COPD overlap syndrome, or ACOS, which is smushing two symptomatic or two syndromic labels together, making a third, even worse, defined syndromic label. But here, COPD, again, it's not just the fact people are breathless, people are dying of these diseases. So, with that context, what did I do? So, during my PhD, and as Tom's already mentioned, my supervisors here were Professor Sir Peter Barnes and Professor Rob Newton. At the time, they weren't Sirs, and Rob was only a postdoc in the lab, and I was his first PhD student. And what I was trying to do was understand a specific pathway within the lungs that led to asthma symptoms. Specifically, I was looking at this cell here called the eosinophil, which is a granulocyte. It's an immune cell, white blood cell, packed with lots of granules that damage the airways when activated. And this cell's development is controlled by a specific messenger molecule, a cytokine called interleukin-5. And this interleukin-5 is mainly produced by T cells. T cells are basically a smart missile of the immune system. They are very focused. They only respond to specific stimuli. And this is actually a good metaphor for my first PhD because I was highly focused. This was what I was focused on. How does interleukin-5 released from T cells, and more importantly, how is it regulated by the drugs we use to treat asthma? So at the time, it was really the blue inhaler, a bronchodilator called salbutamol. So this is relieving that twitchiness, that bronchoconstriction of the airways. There was also a third inhaler called, it's an antimuscarinic, so ipratropium, which was also a bronchodilator. But the mainstay of treatment in asthma at the time really was the inhaled corticosteroid. So this is a steroid molecule, and what this does is instead of acting as a bronchodilator, pardon me, it actually acts to prevent all of this underlying inflammation. It's one of the best controller medications for helping to reduce asthma symptoms and asthma exacerbations. And I learnt a lot. I learnt a lot about pharmacology by looking at these drugs. I learned a lot about immunology by looking at how these drugs impacted on these cells. And I had lots of good mentors along the way during my PhD career. Dr. Duncan Rogers, Dr. now Professor Louise Donnelly, Mark Lindsay, and Mark Gimbich. Mark was particularly useful because he was the pharmacologist in the group, so he helped me construct a lot of my dose-response curves. So that was what I was trying to do at the time, looking at these drugs to try and really focus on this molecule to try and reduce that eosinophilia in asthma. Subsequently, we now have drugs that specifically target this molecule. We have antibodies that will target IL-5 and its receptor reducing the amount of eosinophilia in the asthmatic airway. Unfortunately, the first drug trial of these novel drugs came 6 months before I was due to submit my PhD and showed they didn't have any effect, which suggested that IL-5 had no role in asthma. It subsequently emerged, and we've all learnt a lot along the way, that what we need to do is target the right drugs to the right people. And this was an exercise in not doing that, because these specific anti-IL-5 treatments were given to all comers. So now, and we've learnt a lot, and these drugs are now in clinical use. And although I feel as though I was involved, obviously at the time I was quite upset. But that's what I was doing in asthma. But at the time, this was a respiratory medicine group, and we were also looking at COPD. So now I'd like to introduce you to today's COPD patient. This is Jim. He, as you'll notice, is not eating, unlike my first picture. Jim was born in the early 20th century. He grew up in the 1930s and met his wife at the youth club. Unfortunately, they didn't get married straight away, one, because she was 14, and two, because he had to go away to war, because he was conscripted into the army and fought in Alexandria. He did not enjoy that experience. However, when they got back, they did get married. They had 2 wonderful children. But in 1970, Jim was diagnosed with COPD. Because for the last 10 years, he had been suffering lots of breathlessness, especially breathlessness on exertion. And he also tended to have lots of cracking coughs during the winter. So he was diagnosed with emphysema in 1970. And as I've already said, this is a disease mainly associated with smoking. He was quite lucky. He was able to give up smoking as soon as he was told, "You've got emphysema, please give up smoking." He went cold turkey and did. His wife, Ethel, on the other hand, took another 6 years to give up smoking, and she found it incredibly difficult. So, the root of all evil in this particular disease really is tobacco smoking. It's an innocuous-looking plant, really, but it's native to the Americas and was used quite a lot in shamanic practices and was part of the culture certainly within the Mayan period and possibly earlier. And there are even more archaeological evidence for earlier use of tobacco. 1492 happened. Christopher Columbus discovered the US. And Walter Raleigh was the main person who's associated with bringing back tobacco from the Americas. Apparently Elizabeth I quite liked it. James I hated it, called it loathsome. But up until the beginning of the 20th century, only about 0.5% of the population would have smoked. At the beginning of the 20th century, machine-rolled cigarettes were introduced. So they packaged them much more effectively at much cheaper rates, meaning more people could afford them. And lots of people then took up smoking because it was made to look glamorous. I mean, Audrey Hepburn's an icon, but this particular photograph doesn't help when we're trying to stop people smoking. And indeed, more doctors smoke Camels than any other cigarette. This was heavily promoted by all sectors. I mean, and don't get me started on the ones where there were asthma cigarettes with menthol in them. But then, the UK Doctors Survey actually showed that smoking caused lung cancer. And this is one of the first graphs from these studies showing that, and this is all in men at the moment because smoking wasn't ladylike, although obviously women did do it because Audrey Hepburn did. So, you'll notice that as cigarette consumption went up, so did lung cancer, and cigarette consumption in this country reached a peak in 1965 and has been slowly going down ever since. But what you'll notice is the 20-year gap between the onset of all of that increase in smoking and that increase in the incidence of lung cancer. So when people say to me, but vaping surely better, I say, well, we don't know yet. We haven't been doing it long enough. Okay. If you want to get me on vaping, see me afterwards. So Jim was diagnosed with emphysema. What's emphysema? Well, it's basically where you lose the structure of the lung, and you especially lose those alveolar spaces, those spaces that are necessary for that gaseous exchange. And what you actually end up doing is having large bubbles. You lose those air spaces, they consolidate, and you get just a big air space, sometimes filled with air, sometimes filled with mucus. So up until the mid-1950s, the only way to really study emphysema was in cadaverous patients, so you didn't really know if you had emphysema or not. However, with the advent of better X-ray technologies, and certainly by the time we've now got high-resolution computed tomography, or HRCT pictures, we can show that there are these enlarged air spaces within the COPD. I've been talking for a little while, and I think I need some audience participation. So, if you'd all like to sit up straight. You don't need to stand up, you can just sit up straight. It's fine. I mean, you can stand up if you want. And everybody take a deep breath in. And let it out. Now, I'm hoping for most of you that didn't take very much effort. What you've got within your lungs are proteins like elastin that, they do what they say on the tin, they're elastic. They will spring back into shape. So, that didn't take any effort to breathe out. COPD patients have lost a lot of that elastin, and what they get is air trapping. So, let's see if we can simulate that. Everybody take another deep breath in. And let a little bit out. Now take another deep breath in. And let a little bit out. And another deep breath in. And a little bit out. And another deep breath in.
We get the point.
Did that feel comfortable? No, it wouldn't do, because you've got air trapped, it's not going anywhere, it's not helping with gaseous exchange, you're not getting rid of any carbon dioxide, it's really difficult. COPD patients feel like that all of the time, especially those suffering from emphysema and air trapping. And that's what gives them this barrel-chestedness. They're hyper-expanded. This is the classical presentation, barrel-chestedness with emphysema, and indeed Jim had that. So that's emphysema. We're going to leave that there for a moment because I got a PhD. Woo! And the first thing I did was go to the US, because at the time that's what everybody said you should do. Go and do a postdoc in the US. So I went to Weill Cornell Medical College, based on Manhattan, and apologies to my wife, but despite having our very small studio apartment, I'm there standing in the kitchen. That's a very small studio apartment. Despite that, strangely, people still wanted to visit us. So we had lots of visitors, and this is my brother-in-law who came over for the first visit. It was a wonderful time. We made lots of friends, and these are specifically Licia and Maurizio, who hopefully will also be online. So we had a wonderful time. What's going on? What are we going to do next? Unfortunately, this happened. We sort of changed our plans after that and decided that perhaps we shouldn't make a life in America. And certainly New York was great, but I wouldn't want to raise any children there, especially after this particular world event. So, brilliant. My wife, as always, is brilliant, and she got a job in Birmingham, so we came back to Birmingham. I, however, despite applying for at least 25 positions, I tried to count them on my laptop earlier, because I've still got all of the applications, obviously, because you never get rid of anything, because it's on the computer. Didn't. And I spent 6 months unemployed. Worst 6 months of my life. And my wife will very tell you I was a nightmare to live with. So thank you for putting up with it. What do? Is the world trying to tell me that I shouldn't pursue research? Is the world trying to tell me I should go and do something else? Like, I don't know, I like reading. I applied for jobs at places like Waterstones, for example. What is it I want to do? Do I still want to do this? Again, possibly after 1 or 2 glasses of wine, I decided yes, it definitely was. And I was quite lucky then to get a postdoc at the University of Leicester with this person here, Professor Lerm Ziegler-Heitbrook. And there I started to study the macrophage. Macrophage, we get on very well, we're both big eaters. That's literally what the name macrophage means. So, looking at the macrophage. Now, the macrophage is very different to the T cell. It's not as hyper-focused, because a macrophage needs to be able to respond to whatever's put in front of it. It's an innate cell. It's there to not only get rid of the stuff in front of it, but also to quiet down the immune system after that. It has regulatory properties as well. So despite being known as the guardian of the immune system by basically being the housemaid, it also has these regulatory properties, and this in particular was an anti-inflammatory signal that was released from these cells, and I spent 3 years looking at the mechanisms by which the specific anti-inflammatory cytokine, this interleukin-10, was regulated. And I actually showed that for the first time that it actually regulates itself. So the more interleukin-10 you've got, the more regulation you've got, the more IL-10 you can produce. So it's a virtuous cycle in that particular regard. During that time, I published 8 papers. I know. I was shocked as well when I counted them up earlier. So what do you do then? Well, you apply for grants. But just because you apply for grants doesn't mean you get them. So despite applying for lots of grants during that time, I was unable to continue my employment at the University of Leicester. So, another fork. So what am I going to do now? Well, fortunately, there was a job in the University of Southampton in respiratory disease. Oh, hello. Something that I might know about. Something that I might be able to pass an interview for. But it's in Southampton. I live in Birmingham. My wife has a job in Birmingham. I have a 2-year-old daughter in Birmingham. We've bought a house in Birmingham. I mean, you can already see where this is going because I'm standing here in front of you. But this was one of those other situations where a choice had to be made. And obviously this was in conjunction with my wife as well. It wasn't as if I imposed it. Although sometimes she looks at me, I can see it. We decided that yes, it was worth the move, despite it initially only being for a 2-year contract. So we migrated south, where I was very grateful to be mentored by Professor Djukanovic, who's sitting in the second row there. And again, this was to look at the role of macrophages within these airways diseases, within asthma and COPD. And he's since confided in me that one of the reasons he picked me for the job was because I was the only person who was going to be looking at macrophages in his group, whereas everybody else had their own specific cell type. I had the macrophage. So it's always been my special baby. But in this particular case, we weren't looking at it from blood anymore. So in both my PhD and that first postdoc in Leicester, I was looking at immune cells, so white blood cells, from human blood. Now it's about looking at it from the lung. So how do you get macrophages out of the lung? Well, you can use sputum, which is what you produce when you've got a lot of mucus and a lot of cells within your lung. This sputum itself is coming from a COPD patient, and it's green. And if it's green, it doesn't mean there's lots of bacteria on it, although it might. It just means there's lots of neutrophils in it, so another type of immune cell, because it's the enzymes that come from these neutrophils that make it green. Another way you can do it is to use bronchoscopy. And you can just see me in the back of this photo, ready to receive the samples when they come out of this gentleman's chest using this bronchoscope. Once you've got these cells, what did you do with them? Well, you could look at them specifically, look at the proteins expressed on their surface using a technique called flow cytometry. And I spent lots of hours playing with this specific machine to try and understand what was going on with these cells. And also you could then also look at what genes they expressed, what things were switched on in these cells, and what differentiated them between health and disease. One of the things that was very evident early on is that when you got washings from people's lungs who were healthy, you got a nice clear fluid out of them. If they were a smoker, however, this was the colour of the bronchoalveolar lavage that came out of their lungs, which was slightly annoying because it also made the macrophages that you recovered highly fluoresce, which means that the flow cytometry wasn't particularly good. But during that time I was very lucky and I got my first research grant for me as the PI from Asthma UK. So it was great. It wasn't just 2 years anymore, we've got 3 years. Okay, so we're going to be here 5 years. Okay, should we buy a house again now or not? But around this time, there was, you may remember, the swine flu outbreak. And so we started to look at what role influenza played and how influenza interacted with the lungs. And this again was the first project where I worked with Tom and also another colleague, Ben Nicholas, who also shared that very small office in Ratco's group. But here we showed that we could take bits of tissue from the lungs and we could infect them with this virus. And we showed that this influenza virus not only infected the epithelial cells, but also affected those macrophages. This also led to some more money. We won a prize from the British Medical Association This is me looking very smug. But what we went on to show with that is that what happened in COPD in response to this infection, and this was with my first PhD student, which is Cambridge, we showed that the macrophages were less able to regulate their immune system. They were less regulatory because they didn't express as much of a specific regulatory protein called PD-L1 or programmed death ligand 1. And as a result, they produced more inflammatory cytokines like this inflammatory signal here, interferon gamma. But that led us on to looking at exacerbations. And so exacerbations in asthma and COPD, they're almost as bad as calling them asthma and COPD. Exacerbations are only worsenings of symptoms. So you have increased breathlessness, increased sputum production. It doesn't really tell us what's going on. So, and this was work led by Tom and in conjunction with GSK, but here we were looking at a population of COPD patients for 2 years and looking at their sputum every month for 2 years, and not only every month, but also whenever they exacerbate. I saw an awful lot of sputum in those 2 years. But, as you may expect, what we saw was that there were more exacerbations in the winter. But that means that COPD exacerbations are driving some of the winter pressures that we're still experiencing within the NHS. Some of these were driven by viruses, but a lot of them were driven by this specific bacteria here, this non-typable Haemophilus influenzae, or NTHI, because it's a lot easier to say. So, COPD patients were associated with colonization with this bacteria. They had exacerbations as a result of this bacteria, and also there seemed to be some interaction between viruses and these bacteria. So we didn't really understand that at the time. When Jim suffered from an exacerbation, he referred to it as coughing up blocks. He was extremely breathless. He produced an awful lot of sputum, and really, his quality of life was impacted significantly. Unfortunately, Jim died as a result of a COPD exacerbation. But my research was really looking at how these exacerbations impact on the lungs of COPD patients. Why do we have this impact of exacerbations on the lungs? So we started to really look at not only the macrophages, but other cells that we were already looking at. So back to the T cell. So macrophages, we know, can present antigen to T cells. We already knew that influenza infected these macrophages. But what we didn't really know was how the bacteria were involved in these exacerbations. So, this was a particularly purple patch in our research where lots of PhD students helped us try to understand at least some of these pathways. So we had Grace Cooper, who was looking at the interaction between NK cells and influenza. We had Andy Hutton and Josh Wallington, who were looking at the impact of the bacteria on T cells and macrophages, respectively. Jodie, who's also in the audience, is now looking at the impact specifically of the bacteria when they reside within the macrophages in the lung. And I've already mentioned Richard, and Alistair was also looking at the impact of influenza on the macrophages. So this was a very productive time. It was also a time where I was doing an awful lot of teaching, so I apologise to all the PhD students now because I wasn't spending as much time with them as I possibly should have. But education, and as Tom's already mentioned, bringing on the next generation has really been something I've been very proud of during my career. I actually started my education while I was at the University of Leicester with Professor Derek Rain, who was very involved with an interdisciplinary science degree, and I convinced him to run a module on forensic science. This was all delivered by problem-based learning, and it's probably the reason that when I arrived in Southampton, Dr. Ben Marshall liked it, because that's how the BM4 is taught, via problem-based learning. So I got some teaching experience on BM4. On BM5, I started to co-lecture with Professor Sarah Ennis. But the two people I'd really like to highlight for being mentors and really encouraging my education career are Dr. Jane Warner and Professor Jude Holloway. They have been significant mentors, and they've shown me that if you do it properly, teaching is not a burden, and it should never be really thought of as a burden, but it's really bringing on those next generation. Tom mentioned the ugly word legacy earlier, and I was quite shocked by it, but yeah, if there's any legacy that we can have, it's about bringing on the next generation, not just PhD students, but also our undergraduates. Ramesh was my partner in crime whilst we solved allergic airways disease. I say solved. We evolved the allergic airways disease module, and I was very lucky to then join the pharmacology teaching team, including Professor Tony Sampson, Dr. Emily Swindle, and our unfortunately departed Dr. Tim Miller. So, this was great. We were doing great research. I was doing lots of teaching. Fantastic. Then something happened. So, these are the COVID years. You may recognise some of these from various newspapers. And I was on Sky News. Why they ever want you to point at a coloured liquid? I don't get it. I mean, I know all we do is move little bits of fluid around, but, it doesn't really capture what we're doing. Anyway, so we were showing we were doing stuff, and I'm very lucky to have been involved with Chris McCormack, Rod Hervey, Morella Spaluto, and Vicky Humbert, who really got us off the ground within the CL3. And I should also mention Jenny Russell, who was the technician who kept us on the straight and narrow to make sure we did what we should do within the containment level 3 laboratory. Containment level 3 laboratories on screen look really fancy, but they don't really capture the backache, and the sweaty hands, and the fact that those really big gauntlets are one size fits no one, and you're forever knocking things over. And what we were doing was we were looking at the virus and seeing if we could use some of our extant models, those models of influenza infection, to understand this virus. And what we looked at was looking at a lot of these sorts of assays, these plaque assays, to work out whether we were preventing or increasing the amount of viral shedding from these molecules, from these models. This was also quite productive, but I can't help but feel that we could have done more. One of the reasons I think we could have done more, because unfortunately Ethel was one of the first victims of the COVID pandemic. One of the reasons I also think we could be doing more is because the impetus that COVID gave us, especially for respiratory disease, has not been maintained. It was in the Office for Life Sciences strategy up until the most recent iteration, and now it's no longer an Office for Life Sciences mission. It's respiratory really has fallen off the radar. So what we need to do is really need to bring it back to show that what we're doing is important, because we already know it's important, because people are dying. It's the main cause of winter pressures in the NHS. So the things what we need to do, and the things we think we can do, because we've now got new vaccines against new viruses, And we can we can try and prevent some of these diseases by using that targeted population level interventions. Because we know if you stop people getting infected with viruses, they will not exacerbate. The reason we know that is because we contained everybody in their houses, which is quite draconian and not good for anybody's mental health. But we could show that if people didn't get infected, they also didn't suffer from respiratory exacerbations. We need to be able to better diagnose these diseases so we understand them better, so we can treat them better. And unfortunately, for most people with asthma and COPD, we're still using the same drugs. They may be in slightly different formulations, they may be in a triple therapy rather than in single inhalers, but really we're using the same drugs. We've not had the step change that we've needed in respiratory medicine, really, other than those antibody treatments that I mentioned earlier. And what we need to do is make sure we target them appropriately. How do we do that? It's not just the wine. I promise it's not just the wine. But what we need to do is we need to bring the respiratory community together. And I'm very lucky, as Tom mentioned earlier, I'm the chair of the British Association for Lung Research, which means I'm also in contact with people within the British Thoracic Society, the European Respiratory Society, and it's about now having those conversations, about bringing us together so we can have that impetus, so we can all speak with one voice. And also have wine. But this is about having a network and knowing people. Don't be afraid to talk to people. I think scientists sometimes don't think they're allowed to talk to people. But I think talking is the only way to get over any of these problems. So, I'm coming to the end, you'll be pleased to know. I can see you getting restless. But I was asked what advice I would give my former self. Well, my first one is that a PhD is great. It's a license to travel. I mean, sometimes only between Birmingham and Southampton, but you know, we do. We get to go to nice conferences too. I have painted a pretty dire picture. I do apologise. I shouldn't have. We have had step changes and we have had these new monoclonal antibody therapies, but we now need to see impact beyond those therapies. So there has been progress in respiratory research, but we still have an awfully long way to go, which is great because it means we're going to be in a job for a little while longer. But unfortunately, that does mean more people are going to die. For those people in the audience who are still not put off by my research journey and still want a research career, make use of your networks. Don't be afraid to speak to people. Make sure you go to conferences. Just make sure that you are making use of them. The people you talk to will be reviewers on grants, they will be reviewers on papers, but they are also potential collaborators. So don't be afraid to, in the vernacular, tap them up. Watch out for all of the forks in the road. Only you can make the decisions to go down one path or another. Just make sure it's the right fork for you. And make sure, most importantly, you always have some fun along the way. So, I didn't have pictures of everybody throughout the years, mainly because the hard drive I had with all the pictures on it is very temperamental and I couldn't get some pictures off. But I'd like to thank everybody. I mean, I've tried to thank everybody we've gone through, but all of the PhD students, all of the people who contributed to the ARIS study, all of the people that, you know, go places and wear silly hats with me. And they give me an excuse to dress up. Obviously, I can't do any of this without my darling wife. And the person she's sitting next to me is my darling mother. Hello, Mum. And of course, I can't ever get away with the fact that I have two gorgeous children. Fortunately, they obviously get their genes from their mother. But this talk is really dedicated to Jim and Ethel. They were not only fabulous grandparents, but they were also very understanding landlords when I lived with them whilst doing my PhD. So, I hope that hasn't put you off Science for Life. Thank you all very much for your attention.
Well, Karl, I think it falls to me to say a few words to summarise what a splendid inaugural lecture that was. Thank you very much indeed. I think you've shown us what a balanced academic is all about. So I think that's really, you've brought every element of leadership, research, management, and I know that you have interest in enterprise that you haven't necessarily brought forward there, but I think you only had an hour. Another time. I think that whole culture change piece came through very clearly in how much you care about and look after your students, and I think that that's a really a hugely valuable and valued thing in our academics these days. So it's always very, very interesting to hear about people's journeys, learn something new about science. We always used to talk about lumpers and splitters, and I had no idea that you were creating new diseases as well with all these respiratory conditions. And I also think you've shown enormous resilience in all of those points in your career when you could have given up and gone and worked in Sainsbury's or something. and I suspect many people don't experience that period of being unemployed for a period of time, and I think that is very, very testing, and well done to your wife for supporting you through that. But I can imagine that's why she said you would come to Southampton with the drop of a hat, because that's a hard thing to do. And I guess the other final thing that I noticed was we do a lot of wellness breathing, and I had never experienced that in the foreseeable field before, so I think that was a learning curve for me as well. So thank you for sharing your journey and your excellent life choices, and thank you for your inaugural lecture.